Okay, thank you for the introduction, and we'd like to thank Ari for the invitation, and good morning to everyone. So here, just trying to give you a summary of the discussion we had in London earlier this year on this endpoint workshop, and just to let you know that the summary of the workshop will be published very soon in Journal of Hepatology and as well as Hepatology. Well, as you all know, I mean, we are trying to achieve a functional cure of HBV infection with finite duration therapies, where we would achieve HBs antigen loss, and which defines a functional cure. And there are obviously other type of cures that are more ambitious that we may reach at some point, but we need to, including complete cure, where cccDNA would be eradicated, and even sterilizing cure, where all HBV DNA would be cleared from the liver. But there was, I think, a consensus during that meeting that the functional cure should be the priority now. There were a lot of discussion regarding the terminology of functional cure, whether this is a good term or not. But in the end, even after asking patients' association that were present during that meeting, and with a discussion between academia and industry and even regulatory agencies that were present during the workshop, finally it was decided that there was no better term today, at least, and that we should keep on with functional cure unless someone comes up with a better idea. But just to remind the audience here that the terminology functional cure came up from the HIV field. So the functional cure is defined as a durable HBs antigen loss with or without S antigen seroconversion associated with undetectable serum HBV DNA after completing a course of treatment. It was recognized that it's really a very pragmatic definition that in the end, cccDNA is still present in the liver in very small amounts, and that integrated HBV DNA may be also present, and that HBV reactivation may occur in these patients either spontaneously or during immune suppression. Well, in any case, it was also very important that the patient as representative was there, because it was really stated strongly that the HBs antigen loss is an important goal because it removes the stigma of HBV infection in these patients. So there were all the type of cures that were discussed. I just want to highlight here the realistic functional cure that you all know here, where it is really important because it is we have data now from cohorts, large cohorts of patients, that functional cure is associated with a better clinical outcome in terms of fibrosis progression, as well as the HCC risk. Now, there was a lot of discussion on whether we should stop NUCS or not, and whether stopping NUC would be a control group in clinical trial design. So I don't want to come back to the very nice discussion and lively discussion we had, but really this workshop also had the same type of discussion, and we had a review of all what was said earlier this morning, showing the heterogeneity of the studies in terms of patients' characteristic, criteria for retreatment, and so on. And in the end, I mean, there are only very few prospective studies, as Pietro told us this morning, and they have, in terms of HBs loss rate, they have conflicting results. So it was agreed during that meeting, and the regulatory agencies were there, that to have a stopping NUC arm as a control group would need more robust data because here we have to be very careful. Here it would mean that this would serve as the reference for registration. So that's something that is not easy, as we heard this morning, and there's no consensus on what should be done in terms of treatment cessation. And there's also the fact that we would expose also patients to some reactivation and flares as well. So here it was, the jury is still open, and we really need more data on these stopping treatment strategies. Now, just to introduce the fact that, and that will be discussed later on during the day, that we've learned a lot in the recent years on the mechanism of viral persistence, both on the immunological part and on the virological aspects, which has led to some redefinition of the different phases of the disease that you all know. I mean, they came up in the EASL guidelines, but also led to, I think, better understanding of the viral targets and immune targets for future drug development. And now we come up with quite some very interesting targets. I won't go through this in detail. You will hear a lot during the day. Either direct antiviral approaches that may lead to a cure of the infected cells, hopefully, and immunomodulatory approaches that may also have some potential effect to cure infected cells without killing them, but also some adaptive immune responses that may lead to a specific hepatocyte killing. And now we come up with quite a few targets that are now in clinical development, and you see here a review that was just published now in Nature Drug Reviews Discovery, with tables showing the novel therapies that have been investigated in humans. So there are quite a few, and we are now in this situation that we remind us the HCV field a few years ago. We have all these assets coming to phase 1B, phase 2A, and we know that at least today, we need to combine different mode of actions, but we don't know how to do that, with which type of regimen and so on. So here it's reason why it's very important to have this type of meetings here to discuss what would be as a rationale for the best combination in the future. So having said that, I mean, we have also to remember that we are dealing with patients that for whom their liver disease can be controlled very efficiently and in a very safe manner with nucleoside analogs. So for whatever new therapy is coming to clinical development, assessment of safety is really a priority. And so the bar is very high because all the new therapies will have to prove to be at least as safe as a NUCS currently, and this come also to the first question on stopping NUC. If we have in clinical trials stopping NUC arm, we have to be careful that we don't expose patients to a risk of reactivation and flare. Now, obviously, if additional risk is anticipated with different mode of action, it would have to be well justified for the endpoint that is achievable. So that has to be discussed on a case-per-case basis. And one of the major concerns in HBV drug development is the occurrence of ALT flare. So here you see a definition that came up during that workshop, and you see it's different from what you heard a little bit earlier. So everyone comes with his own definition. So during that workshop, I mean, and there was quite a lot of different perspective in the audience. I think there was kind of a consensus to distinguish three types of flares, so the antiviral flare, the virus-induced flare, and the drug-induced flare. So one of the very important points from Marion Peters was that with all the type of studies that we've seen is that usually we know whether it was a good or bad flare after the flare resolution. Now we have to see how can we anticipate and predict what will be the outcome and whether it was going to be an antiviral flare or virus-induced flare. And obviously, the drug-induced flare is more scary, as we know. So this will be discussed anyway by Harry Jansen, but this was really a very important point of the workshop and should guide, I think, the clinical development of new drugs. So now we are in this situation where we have these different drug targets and assets that either target directly the virus or try to harness the specific immune responses. And the question is, are we going to combine this, in which manner that would be safe and more effective than the currently available drugs? So one of the obvious questions is the design of clinical trials, which was really discussed during that workshop. And there's no, clear guidance in terms of everything should be done in this way or the other way, because we know that the disease is heterogeneous and that depending on the mode of action of the drugs and depending on the patient's group we want to target, the design of the trial may have to be different. So this has to be really thought of really carefully in advance, depending on the drug mechanism of action. Many of the phase 2 trials usually target patients who are virally suppressed because it's an easy population to access. They are homogeneous. The risk of hepatitis flare is less. Liver disease is controlled, and patients are also willing to stop at some point to have improved therapies to stop their treatment. It was also, I think, agreed that the demonstration of superiority would be generally preferred for the HBs loss endpoint, but that the non-inferiority might also be an option if, for instance, we had a situation where sustained viral suppression with a finite treatment would be similar to a lifelong therapy with NUCS. So this has to be also discussed, and we had a very interesting discussion yesterday night. It was a first workshop on that point. Now, in terms of patients population, which patient population to target as a priority? The patients with chronic hepatitis, high replication with inflammation, have high need, and there's a greatest gain for effective therapy. All the others group are also very important. The so-called immune-tolerant patients, because of the long-term impacts that we could have on the disease, and some of the immune-based therapies may also be very more effective than we thought before in that population. The low replication group, as we just said, the NUCS suppressed patients are important because, at least in the early stage of clinical development, because there are less safety issues in that population. All the other groups, like cirrhotic patients, HIV, transplant patients, children, would come really after that. And there were questions regarding stratification, but we have to be really realistic in the early phase development. We have to stratify on a very few factors, otherwise the size of the trial would be much too large. Then there are some new clinical trial design that came up with the oncology field, and now what is really interesting is a platform type of trials, because in that situation you can study multiple targeted therapies in the context of a single disease in a perpetual manner, so that you can have a single infrastructure that is ongoing over time. There's no fixed stopping date, and you can use a command control arm that will aid efficiency. So you don't have to restart all your clinical trial design, and you come with your different targets one after the other, and at some point you may be in the situation where you can combine them. So that has been used very often with in the field of oncology. And I know that some companies are now using that type of platform trial design in their clinical development process. So regarding the primary endpoint and desired response rates, a lot of discussion. So in phase 2 trials, the HBs loss may be still very important, but there were discussion regarding a decline in HBs antigen, whether it's 1 log, 2 log, or a decrease below a certain threshold, so that we would minimize the risk of abandoning promising drugs. Clearly, in phase 2 trials, we need to have early signal of finite treatment efficacy. For phase 3 trials, HBs loss is a clear endpoint. 30% rate is an arbitrary target, but we need to start with something, and this is really needed to guide the first wave of drug development, and then we can refine once the clinical development is progressing. A sustained off-treatment HBV DNA suppression is also considered, because it would be even something better than what we currently have. So this is also has to be kept in mind. The decline in HBs antigen level as a predictor of HBs loss in early phase trial needs to be considered as an exploratory endpoint, not a surrogate endpoint today, because we don't know how to use that marker, the decline in HBs loss, in terms of prediction of the HBs loss. Now, the last point is novel biomarkers. So we've heard a bit in the previous session, and we hear more in the next presentation. We will need to have novel biomarkers to assess target engagement and treatment endpoints. Here I've just shown the viral biomarkers, but we also really need immune biomarkers, and we just discussed that in the discussion. But for all these novel biomarkers, we still need to have standardization and validation. And there was a clear highlight that we need to show that these biomarkers, these novel biomarkers, would predict the HBs loss and would be helpful in guiding therapy. So the last point is regarding combination of new investigational drugs, but I will be very short, because Poonam Mishra will elaborate on that at the end of the day. Clearly, the FDA and EMA are open to discuss a combination with novel investigational drugs, and I will leave this to Mishra later on. And I would like to thank you for your attention, and would like to advertise the HBV Cure workshop from ANRS in France, which will be held in May next year. Thank you.